Good day, dear students. I hope that you all are in good health, and I'm glad to invite you for last lecture of biochemistry this year, which will be devoted to the Crimean Biochemical School. Write please the topic of the lecture, Crimean Biochemical School. And scientific directions of the Biochemistry Department of Medical Academy. In the left column, you can see the coat of arms of our department, and after lecture, you will understand why it is designed especially by this way. Here you can see the immunoglobulin scheme and something which is interacting with it. Founder of the Crimean Biochemical School is a professor of German Vasilievich Troitsky. For almost 40 years, he was the head of the department in the Crimean Medical Institute. German Vasilievich was born in 1913 in Kazan. He began his research activity already during his study in the institute in Rostov-on-Don, and in 1940 he defended his philosophy doctor thesis in medicine. But his life was dramatically changed by the Second World War. He was in the active army and went from Stalingrad to Berlin. He was awarded even with the Order of Red Star. German Vasilievich revealed the Crimean Peninsula from the fascists in the 1944, so it was the first time of his visit to Crimea Peninsula. After the war, Troitsky settled in Moscow and continued his research. Finally, in 1951, German Vasilievich brilliantly defended his postdoctoral thesis devoted to the biochemistry of vitamin A. And after this, he was invited to the Crimea by Sergei Ivanovich Georgievsky. Academician Pavlov wrote that science develops as well as new research methods appear. Another Nobel laureate, academician Landau, formulated more precisely, the method sometimes is more important than a discovery. The important research method to study blood proteins in clinic and experiment was electrophoresis. First electrophoretic device was created by Swedish scholar Arne Tiselius, who was awarded the Nobel Prize for this in 1948. But his device was bulky, and when German Vasilievich headed the department, at first he improved optic system by installing of the special lens, and electrophoresis of proteins became quicker, and this allowed to formulate the new direction for the department study of blood serum proteins. Headed by Troitsky, everyone followed this rule and paid great attention to the invention of and modernization of equipment and techniques for fractionizing of proteins and study of their chemical and physical properties. The experience of work was summarized in the monograph of Troitsky, Electrophoresis of Proteins, published in Kharkov in 1962. Later, the first in Europe spectropolarimeter was constructed by German Vasilievich and his students, device measuring optic activity of protein solution in different wavelengths of light. For the first time, on the basis of findings obtained with the help of spectropolarimeter, Troitsky advanced an idea about existence of the beta sheets in globular proteins. At that time, there was an idea that globular proteins contains only alpha helical structures as a secondary type of organization of protein. First, these findings were published in the magazine Biophysics in 1965. The experiments carried out in many laboratories of the world after this, including X-ray structural analysis, provide this theory, and nowadays the presence of beta sheets in the globular proteins is generally accepted by. Here you can see the part of immunoglobulin molecules, light chains variable and constant domains that contains mainly beta sheets in the globular structure. And here you can see that beta sheets also are the main secondary structure in the heavy chains of immunoglobulins also. Another achievement in methodology was the invention of absolutely new method of isoelectric focusing. Separation of protein compounds is an important task in many areas of the human activity. For example, for the receiving medical substances or for diagnostics of disease. One of the most effective ways for the separation of protein is the method of isoelectric focusing. The scheme you can see here. This method is based on the motion of proteins under the influence of electric field in the solution, with the concentration of hydrogen proteins gradually changing, so-called pH gradient. If electrophoresis will be done in constant pH, here we have motion in gradient of pH. Professor Troitsky was the first scholar in the world who, together with his students, suggested to create gradient pH with the help of variable concentration of polyatomic alcohol, polyols, for instance, glycerin. You know this alcohol that contains 3 OH group, and in buffer solutions on the basis of the boric acid, so borate polyol systems. This method is based on the idea that the more concentration of polyol, the more the concentration of hydrogen protons in the medium, and the higher the density and viscosity of solution. It made possible to generate stable pH gradients for isoelectric focusing. The research results were summarized in the monograph of Troitsky and Kazhitsky, Isoelectric Focusing for the Proteins in Self-Organizing and Artificial pH Gradient, published in 1984. Moreover, this method was used in the space experiment according to the program Tauria for the division of proteins receiving particular pure preparations under zero-G conditions without gravity. These experiments in space were studied in the space station Salyut 7 by cosmonauts Berezov and Lebedev, and then were continued at the orbital complex Salyut 7, Salyut T5, Salyut T7 by cosmonauts Serebrov and Svetlana Savitskaya in 1982. These are photos, historical photos of German Vasilievich Troitsky with cosmonauts in 1980s year. And this is the photo of experiment directly in the space in 1982. This is Savitskaya and Serebrov, the Russian cosmonauts, Soviet cosmonauts. They obtained very pure proteins. For example, some of the proteins that they separated were protein antigens of the viruses. And at the beginning of the 50s, Troitsky predicted the importance of studies of human serum proteins in health and disease. He suggested that any disease starts with the damage of molecules and do affect the main molecules of the organism, proteins. This sort of Troitsky have been developed by his followers and are still relevant. So he found a new scientific direction, so-called pathological anatomy of the protein. And the final achievement of Troitsky was the theory of immunity. The backbone of this theory includes the following. When some endogenous or foreign toxic substances get into the organism, serum albumin takes the fall bounding these harmful molecules yet before the immune system comes into play. So albumin plays the key role in this theory, and in addition, albumin is modified and sends by this modification signals to the human organism to combat dangerous substances as soon as possible. So albumin is the first border for the harmful molecules, and modifications of albumin were studied at many diseases by the command of Troitsky. His dispute with some former managers of institute became a classic. What is more important for the university? To train good students or good professors who will train good students? The answer is clear for anyone. The university that doesn't train specialists for itself is going to fail. To train a good specialist, just knowledge is not enough. The figure of professor plays here a significant role. He must be able to cultivate a love for learning in his students. German Vasilievich was extremely intelligent human and great teacher. And his greatest achievement, in addition to scientific achievements of his school, of course, the training of whole pleiad of scholars dedicated to science for new discoveries. Let's repeat the most important achievements of German Vasilievich Troitsky and founded by him school. The creation of apparatus for electrophoresis and improvement of method. Designed the first in Europe spectropolarimeter, and with the help of this apparatus, for the first time in the world, the existence of beta folds, beta sheets in globular proteins is suggested. A method of isoelectric focusing in Barrett polyol system has been developed. The separation of proteins under microgravity conditions at the space station. Found transition changes in proteins in pathology, especially in albumin, and studying of immunoglobulins. A new scientific direction has been created, pathological anatomy of the proteins, and the theory of brain unity has been developed. Students of German Vasilievich Troitsky formed the Crimean Biochemical School. And here you can see the present head of the Department of Biochemistry, Professor Konstantin Alexandrovich Yefetov. He was a student of German Vasilievich Troitsky, and in his honor, Professor Yefetov organized many conferences and competitions of the school children and students' work, ordered to the name of German Vasilievich Troitsky. You can see the Wikipedia page devoted to the German Vasilievich. Also, some books were published with the sites of German Vasilievich. Book is named How Boss Tells, and also the book with devoted to the memory of German Vasilievich with different memoirs and papers that are devoted to him and his work. Professor Yefetov is a department head since 1999, and the most important direction of our further studies is the study of immunoglobulins, genome and pathology. with use of monoclonal antibodies and spectral methods. Let's list modern scientific direction of research on our department, and then we'll discuss them more detailedly. So the first, it's a study of modifications of immunoglobulins. We put the concentration and our attention more to immunoglobulins modifications, not albumin, in malignant tumors, but also at other diseases to compare how what changes we can find. And we are doing this with the help of spectral methods, especially with the ultraviolet differential temperature spectrophotometry, and also antibodies. So the second direction, which will help us in study of human proteins, is obtaining of monoclonal antibodies to human immunoglobulins, their parts, and also other molecules, other ligands that can be modeled in our researches. We put our attention to the insect proteins, why I will explain a little bit later. And this is the second direction, which require usage of hybridoma technology. Professor Ifeta on his lecture told you a little bit about this. I will remind today also in short. The third direction is the sequencing of the mitochondrial cytochrome oxidase gene. So this is the investigation of nucleic acids of different organisms and usage of this information in different ways. A very interesting direction, model for the immunologists. Then fourth, our direction, it's a study of hemocommunication of biological species and synthesis of the new sex attractants. Also, how it is possible to use, I will tell you a little bit further. Molecular control of the tumor process in paraproteinemic hemoblastosis, very long time. In parallel with the study of immunoglobulins, we are studying the diseases which are accompanied with the changes in secretion of immunoglobulins, and especially paraproteinemic hemoblastosis, tumors from plasmacytes that are synthesizing and secreting immunoglobulins, monoclonal immunoglobulins, into the blood serum. In my lecture devoted to blood, I told you several words about these diseases, and today again remind you and show what we are doing in this direction. So about the first our direction. Immunoglobulins, these are molecules that are the most important in the action of humoral immunity. They are adapter molecules that are interacting with different antigens, and by changing a conformation of the constant domain can transfer the signal about presence of foreign antigens to the immune cells and cause to the complement system, and can cause different reaction of antigens, starting the elimination of antigen or destruction of organisms that have these antigens on their surface. I will remind you in short, you can see the molecules of immunoglobulins, two heavy chains marked with the dark green and blue color, dark blue and light blue color, and with the deep red and light red, you can see two light chains. Each chain have approximately separately compacted parts which are renamed domain. Heavy chain has 4 domains. It's a scheme of structure of immunoglobulin of G class, and 2 domains in each light chain. This part of the molecule is named constant Fc fragment because it is nearly the same in all immunoglobulins in the class. And here we have variable domains of light and heavy chains that are forming antigen binding site. Especially here, the antigens will be recognized. This will change the constant domain. And here in constant part, the complement can be bound Schematically, it was reflected in the coat of arms of our department. And other different things can interact with immunoglobulins, non-antigenic. It means that different amphiphilic phospholipids, it was shown that they all can interact non-specifically with immunoglobulins and can be used as a model molecules to study changes in immunoglobulins pathology. Here you can see the carbohydrate component and glycosylation of immunoglobulins is necessary and absolutely necessary for the normal functioning of these molecules, but this component is present in CH2 domains. Two oligosaccharide branched oligosaccharides are linked to the CH2 domains, but sometimes the different pathology, excessive glycosylation can be found in the light chains, in the other domains of immunoglobulin molecules. I hope that the professor explained to you how with the investigation of temperature differential spectra we can investigate stability of the conformation of immunoglobulins. And this picture you saw already the special value between perturbated residues of tyrosine and tryptophan can be calculated after several experiments with the help of spectrophotometer, and then was found that from immunoglobulins from the blood serum of healthy people show the value of this index more than 1.6, and in people with malignant tumor the stability of conformation is significantly higher, and different perturbants such as urea, which were used for this investigation, cannot make changes. So conformation cannot answer to the stimulus from the environment in people with malignant tumor. And this can be used for the molecular control and diagnostic and prognostic and control of treatment in these malignant tumors. This was a special case of the patient with the tumor of stomach. When he was admitted to the hospital and this diagnosis was verified, the value of our indexes was very lower, significantly lower than 1.6. After operation, after several weeks after operation, it was characteristic for the healthy people. And after a year, when this patient was being admitted to the prophylactic, nothing was found, no tumor, no changes in lymphatic nodulus, but investigation shown that the index again go to the pathological significance. Unfortunately, surgeons did not find anything to treat, and the patients go home and return only after 1 year when the metastatic tumors were observed in many organs and it was untreatable. Operation was not necessary, and patient died soon. And this case shown that this value can be investigated and can show that here it was necessary to investigate more precisely this patient and maybe start to treat him, maybe with chemotherapy, and something must be done to prolong his life. It's an illustration that this method can be incorporated to the clinic and can be used really at least for the control of the results of treatment, results of operation in some localization of malignant tumors. The second direction, as I told you, we need to obtain for the investigations of the interaction between molecules. We have to obtain tools, and these tools are monoclonal antibodies. By the hybridoma technology, with the usage of beta cells from mice, and myeloma cells that are not produced immunoglobulins, also my cells, we can, by special procedure, lead to junction of them, and then on the special selected media leave only cells that obtain from the after immunization, obtain genes from beta cells that answer at the necessary antigen, and from myeloma the genes of tumor, and start to be this hybridoma tumor will be cultivated long time. As we will be food, these cells will live and produce necessary for us immunoglobulins. We are testing all the cells at first and search from them only synthesizing necessary for our antigen immunoglobulins. Then we are cloning them, recloning, and finally obtain the cells that we can cultivate on the medium or inside of the body of the animals. And usually these cells live in the abdomen of the mice. We can take this abdominal fluid which containing antibodies, or tissue fluid, medium fluid, which also contain antibodies. This here you can see the hybridoma box of our department, and Professor Yekveter that is working in hybridoma box in obtaining of monoclonal antibodies. To create model system, primary, I hope that Professor Yekveter told you that the primary idea to explain why these changes in modification of immunoglobulins at malignant tumor, idea was that products of destruction of tumor cells and their membranes, for example, they are amphiphilic. It means phospholipids are amphiphilic, some small proteins and peptides, they are amphiphilic, and they can interact non-specifically with immunoglobulins and change their conformation, cause instability and decrease the activity of these antibodies in the way to interact with normal antigens and interaction with the complement. So this interaction with product of destruction, amphiphilic cells, amphiphilic part of cells and membranes can decrease the functioning of antibodies. Antibodies with lower function cannot normally find antigens of tumor, as tumor cells also loss eliminate. So this is the closed ring and pathogenic one of the suggested pathogenic mechanism of the survival of the tumor cells. So to check this idea, it was necessary to take something amphiphilic and incubate with the normal immunoglobulin and check how it will be changed. Will it changed or not after this? And as a model amphiphilic molecule, Professor Yekveter proposed to use peptide from the bee venom, which is named melittin. This is the model of this small peptide. It is amphiphilic and it was incubated with the normal immunoglobulins. And after this, our value was again measured. And you can see by the arrow indicated the immunoglobulin that was without incubation with melittin. It was in this zone, so value was characteristic more than 1.6, near 2 or more. And after incubation with melittin with amphiphilic peptide, conformational changes transfer it to the red zone. Together, it shows the conformational changes like immunoglobulins from the blood serum of proteins with tumors with pathology. So this one of the confirmation of this idea you obtain. And for the creation of so-called molecular microscope for investigation how molecules interact one to another, the very good method is ELISA, or enzyme linked immunosorbent assay. It is performed in the special plastic supports with many small test tubes, where we can put molecules which can specifically interact. And will this interaction be or not, we can check with the help of another immunoglobulins linked to the enzymes. In each stage, we are washing, after incubation, we are washing the plate, and everything that is not reacting go away. So if finally enzyme will act, it will mean that really something antigen is present in the plate. Here on the bottom of the plate can be antigen, here can be immunoglobulins. It depends from the variant of the immunoassay, sandwich or direct or twisely indirect. After this, the intensity of the color changed by enzyme substrate is measured on the spectrophotometer polychannel, and this helps us to explain how strongly molecules interacting, less they interacting or no interacting completely. As a new model molecule for the investigation of immunoglobulins pathology, we choose lipoferin. Lipoferin is a lipoglycochromoprotein, main component of the hemolymphoma lepidoptera. Why we took insect proteins and peptides? Because phylogenetically insects are very far from humans, and due to this, monoclonal antibodies to their proteins and peptides will not give cross reactions with the antibodies against of human proteins. Moreover, lipoglycochromoprotein lipoferin contains many different components, and this is amphiphilic protein. Presence of non-protein components such as lipids, carbohydrates, carotenoids make them very convenient model molecule, because carbohydrates we can investigate with the help of lectins. Lipids give amphiphility. Carotenoids give color to this protein. Really, it is yellow colorated and simply identified in gel electrophoresis. So this is stained electrophoresis. You can see the stained fraction of lipoferin. But before staining, we can preparatively find it in the gel and take it and evaluate to obtain easy big quantities with a good concentration of this model molecule to use it after this immunoassay. And it is immunogenic because it's a great protein, and immunization of mice will be successful with the help of this protein. So it is really possible to obtain monoclonal antibodies to this protein, which we did on our department. When we obtained such immunoglobulins, we proposed several schemes how to investigate immunoglobulins in the ELISA method. So here you can see the one cell of the plate, and in one variant we can put lipoferin at the first stage, then immunoglobulins obtained from the blood serum of investigated people, healthy people or people with pathology. Against of these immunoglobulins, we have obtained it on our department mice antigens, exactly against of immunoglobulins of their chains, and to show is reaction will be or not, we can use sheep antibodies against of mice immunoglobulins linked to the peroxidase, and peroxide with the special stain we can use to check the result of reaction. Another type of interaction when we can put immunoglobulins investigated at first, and then put on the second stage lipoferin. Lipoferin is reacting not very effectively because it's a non-antigenic interaction, of course, but it was shown that really this amphiphilic protein can interact with immunoglobulins, and it was shown with the help of obtained before anti-lipoferin mice and monoclonal antibody. Here you can see the same sheep anti-mouse antibodies linked to peroxidase. These schemes we used for investigation of degree interaction between lipopharin as a model molecule and immunoglobulins obtained from the different people. I will show you the results of this investigation. Here you can see the level of interaction of lipopharin and IgG from blood serum of healthy people, it's a green color, mixture of donor blood serums. And patients with ulcerative stomach disease without complications or complicated by stenosis. Why we choose especially this pathology? So this is inflammatory pathology, non-malignant, but sometimes stenosis of pylorus change the symptoms of the ulcer to the more same with the malignant tumor of stomach. And here you can see that there were not significant differences, so possibility and mistake was so much that we cannot find any differences between these interactions. But when we start to investigate people with the different tumors of the gastrointestinal tract, with the breast cancer, with the intestinal cancer, stomach cancer, pancreatic cancer, we found that most of them shows more higher interaction with lipopharin in comparison with healthy people. And this is very interesting because it also confirms our theory that immunoglobulins in malignant tumor cannot perform their function because they are already attached, already are taking parts of membrane, something amphiphilic like amphiphilic phospholipids or peptides or proteins. So these investigations are continued on our department. Just now we are again trying to obtain the portions of lipopharin for this investigation and continue to obtain immunoglobulins from the blood serum of the patients with different pathology. We are investigating them by the spectral methods and also with the help of monoclonal antibodies and model molecules. But monoclonal antibodies to animal protein can be used by another way, by the way to compare interactions of these monoclonal antibodies to proteins obtained from the different biological species which are relatives, but to solve their real relationships and maybe pathway of evolution of this group of the animals. This new direction, monoclonal immunosystematics, was proposed by Professor Ifetov, and this is a scheme that illustrates you can put antigen, any protein obtained from different biological species on the bottom of the plate, then use monoclonal antibodies, and if they will react, the force of reaction will depend from the quantity and similarity of the antigens. And you know that proteins are coded in our DNA, in our genes. So evolution of the DNA of genes is reflecting in the evolution and changes in protein structure. And as more nearest species you will take, as more nearest values of interaction of these monoclonal antibodies you will obtain. And with the help of this scheme, we investigated relationships between different representatives of Lepidoptera of the family Zyginida. It's our model group of insects that from which we are obtaining these proteins and peptides for the study of immunoglobulins. So this is the schemes for interaction of monoclonal antibodies with lipopharin from different biological species. And after calculation of the value of interaction, we can obtain so-called dendrograms that illustrate that, for example, these two species are more nearest one to another than these two. And for example, this can form one group of species and this another group. And they are separated by the special value of the interaction, and this can be used for correction in biological systematics of this group of animals. Another direction is the study of DNA itself. Sequences of the mitochondrial cytochrome oxidase genes in some nuclear genes in different biological species. Here you can see the model of the cytochrome oxidase protein from the respiratory chain, if you remember, yes. So this is, it is coded by mitochondrial genome, especially we told you that not all proteins of mitochondria are coded by mitochondrial DNA. Only some of them, nearly 5%. All other are coded by nuclear genes. And as it was shown, especially nuclear genes cause enzymes of citric acid cycle, and sometimes it is necessary to study them really to obtain species differentiation in the study of animals. The gene that is coding first subunion of cytochrome oxidase, which is CO1 marked usually, contains nearly 658 base pairs, and sequencing of them and comparison gives good information for the biologist, systematics, and even allow to create the special passport, so-called barcode, for each of the biological species. It can be convenient, for example, when it is necessary for medical, for criminal investigation, for biological investigation, to determine biological species in the not whole body of the organism, or at different studies. For example, any biological study of the organism development can be identified by DNA. And it was shown that especially this piece of the mitochondrial genome really brings great information about the biological species. In these times, Department of Biochemistry heads the international scientific project on DNA research. It is named ZIGNOA, devoted to the DNA most, carried out jointly with the University of Guelph in Canada, as well as together with researchers from other countries like Austria, France, Germany. They help us to collect material, and then after correct collection and documentation, this material will be sequenced in Canada by the special equipment and elaborated for this methodics. This project is present in internet. There is a special site which shows how many specimens we did. Look, it's a great quantity. At this moment, it's more than 1,235 specimens, and it is a list of museums and localizations where we can, from where we took the material. So this is not only from Crimea. You can see that from over the world, this material is following for us to be collected and then to be investigated and analyzed in the view of this, in the frame of this project. Here you can see our Austrian friend, Professor Gerhard Tarman, that is helping us to collect the material and to put it in special plates that after will be sent to Canada for the sequencing. He is coming to Crimea nearly each year and also deliver the lectures for our students and teach us very often. Unfortunately, this year, due to the epidemiologic situation, it was not possible, but if he will come next year, please, you are invited to his lecture together with Professor Viketa. It's very interesting information and communication with such a great scientist is very useful for the doctors, for the physicians, and maybe scientists. In Canada, the sequencing of this piece of mitochondrial genome is performed, everything is computerized, and then data are available for the analysis, for the calculation of relationships between species and species identification. Sequences will be done and the result will be obtained as the density, and which will recognize each nucleotide at in its position. You can see they are colorated with different colors: thymine, adenine, cytosine, guanine, and numeration of nucleotides will be available. And then for each specimen, we have the special page where the total sequence is represented. All data of the specimen are represented. There is a photo if possible, and barcode. The color barcode is also represented here. You can see that this is the passport of this specimen, and in the specimens of the one species, the differences are minimal. Inside of one species, in most of the biological species, differences is not more than 1–2%. After this, with the help of special analytic programs, you can analyze all specimens or part of them as you want, and create different dendrograms that will show you how really what is the difference inside of the one species, and what is the difference between the relative species, calculate distances between different groups of the relative species, and then. This will help in the discovery of new species. It will help in zoogeographic investigations, and then many tasks can be solved with the help of this method and usage of this sequencing technique. One of the results I can represent you here. Look, these are by points. Here are indicated places from the origin of different specimens that we studied by this technique. From Italy, you look from the Crimea, from the Ade, and here under the Caucasus, here in Ukraine. And finally, we found that these representatives of one species shows a little bit different barcodes, and we can separate them into the two groups that, as we know now, has a little bit different origination and evolutionary history. So this is one of the biogeographic tasks that was solved with the help of this method. One more direction of our department is the study of hemocommunication of the species, because hemocommunication is very important for the development and then for the existence of the species. And this hemocommunication will be performed with the help of molecules. We biochemists, we are studying molecules, and especially with such an important biological function like sex attraction. You know that Crimea is not only a place for recovering, but also place recovering and rest, but also place where with very good developed agriculture. And one of the direction of this agriculture working is the cultivation of vineyards and obtaining of grape, and then creation of different products of it, especially wine or juices, and then. So big quantity of area of the Crimea are covered with vineyards, but unfortunately there are pest species that are problems for the agriculture, and one of these problems at the beginning of the 20th century was a larva of special zygomate moth, Terazimima ampelopaga, in Greek ampelos it's a grape, and paga eating, so eating, wine grape, yes. Look, this larva completely can destroy the leaves of the vine, grape one, and it will decrease the final result of the vineyard significantly more than 50–70%. The active usage of pesticides leads to the disappearance of these pest species from the vineyards nearly after 50 years of the 20th century. But you understand that this require several times to work with the very toxic compounds. Even workers are protected from it because they are really very toxic, not only for the pest species but for the human also, and of course it decreases the ecological properties of the final result, final wine grape, and do the worst for ecology for our recreation area. So people are coming to the Crimea for the rest and recovery, for reconvalescence. They want to breathe with the clean air. They are going to the sea, but great areas are blocked with the pesticides, and this decrease ecology, of course, decrease the possibilities of our recreation resource. Unfortunately, at the end of 20th century and beginning of the 21st century, the Terazimima ampelopaga was again found not only at the perimeter of the last bushes of the vines in the special vineyards, but also it migrates to the decorative vine leaves which are used in the different our culture area, different customs in the castles of our beautiful Crimea Peninsula. Of course, here we cannot use any pesticides, and actually this is a reservoir and depot of Terazimima ampelopaga that in each moment can go back to the vineyards. What to do? One of the way to decide this problem is to create ecological, non-toxic methods of fighting with these pest species. It was shown that for the attraction, the males of Terazimima ampelopaga use the special molecule. The structure of this molecule was determined, and this is 2-arbutyl 7Z tetradecyanated. So this is the ester of the one type of the alcohol and non-saturated fatty acid with the double bond here in the 7th position. We proposed that molecules with the similar structure can also be active in attraction of this species, and take another a little bit shorter fatty acid dodecyanated and butanol to create ester between them. But position, of course, of double bond is not 7, it's a 2. We obtained the structure that is named 2-butyl dodecyanated with the special help of chemists from the Crimean Federal University. We use this substance to check in field will be it attractive or not. And fortunately, it was attractive, and we put this substance to the special stick traps for elimination of the males, and by this create the methods of protection from this pest species. With the help of these new artificial sex attractants, we did the monitoring not only in Crimea and not only for Terrasinoma ampelophaga. It was attractive also for other species. Here in Crimea you can see the attraction of the two species, Terrasinoma ampelophaga and Yardanita subsalana. It's another related species, but not pest. Anyway, you can see that with the help of this new sex attractant, it is possible not only create the methods of fighting against this pest species, but do the monitoring of the flight period of this species or other species. Sometimes to find species in the special areas, so new biotopes can be observed. And if attractants are active for example, rare species, we can find the zones that is necessary to protect, and then many usage of this direction we can name. And of course, this is related to prophylactic medicine, because allow us to decrease the quantity of pesticides that can be used in our vineyards, and by this way increase ecology and increase the health of our people and people who are coming to the Crimea for recreation, for rest and reconvalescence. Here you can see that these sex attractants now are tested in different areas of Russia, Italy, Austria, Laos, and some different countries. And look how many species were attracted to this, and modification, further modification of this molecule. We created already several variants of molecules of sex attractants, and they are tested over the world, and helps to many scientists to decide and solve different tasks of biogeography, of evolutionary biology, and then. So this is very good work. There is a request for our sex attractants, and now we are on the way to obtain new molecules and testing them in the fields, first in Crimea and then in other countries. We are not put attention only to the chemical features of our objects, but also doing everything that we can for the study of morphology and cytogenetic of these species. Different morphological characters are studied in our department. It's a heterotaxy position of seta on the larva of this organism, these inner organs, and cytogenetically we can investigate the karyotypes of this group of animals. So we are using them, but we are also studying them as more as possible and increase our knowledge about biology, ecology, and biochemistry of this group of animals. And one more direction that I want to discuss, it's a molecular control of the tumor process in parapratinated hemoblastosis. I told you that there is a group of special diseases, hemoblastosis, monoclonal gammopathies. These are malignant tumors from beta lymphocytes that are localized in our clean bones. Plasmacytes normally are synthesizing and secreting immunoglobulins, but if only one clone starts to divide it many times, all the tumor cells will be daughter cells of the initial of one clone. And of course, they will produce immunoglobulins with the same primary structure, it means same secondary, tertiary, quaternary structure, same charge. And in electrophoresis, they will go to the one zone. In this zone, we will see by densitometry the special M gradient or MP. And in a gut gel, you can see this is the healthy blood serum of the healthy person. Here, gamma globulins are separated and diffused in different zones, not very greatly stained. If paraproteins are present in human blood, in blood serum electrophoresis, you can see MPIC or the special spot, very greatly stained. It can be localized in gamma zone or alpha beta zone. It depends from the specificity of primary structure and charge. And here, for example, this dysproteinemia is accompanied with a decreased quantity of albumin because it's a tumor protein, and of course, it will do some harm for the patient, and also tumor itself will intoxify the patient. In the X-ray examination of plain bones of such a patient, you can see the defects of the bones where tumor already destroyed the bone, and the greatest quantities of immunoglobulins, absolutely the same in structure, we are naming them paraproteins, are coming to the blood serum. Investigation by electrophoresis allow us to reveal them. But we go further, and we wanted to measure how many paraproteins really, how many proteins are represented by these immunoglobulins in the patients, and how this quantity will be changed during the treatment. Here you also see the electrophoresis in gel of agar. Here healthy people, and here two patients with malignant tumor, with multiple myeloma. The group of this disease includes multiple myeloma, macroglobulins of Waldenstrom, and other diseases with malignization. Also can be accompanied with the appearance of an ingredient or paraproteins in the blood serum. After electrophoresis, we created the program that after scanning of electrophoregram can work with the spot, can separate and do the border between the spot and other fractions of blood serum proteins, and calculate the specific index. We named it M-gradient index. If the investigation will be done for the one patient that was treated, we can obtain several data and several significances of M indexes during the treatment. Here you can see that for the model, for the creation of the program, for the creation of correct formula for calculation, we take paraprotein from one patient and solve it progressively, and it was shown that with the very good value of regression, it is a direct line. Using this line, we can recalculate concentration of the paraprotein after the determination of M-gradient index. And here you can see the 4 different spots that are obtained in one electrophoresis and already worked with our program. Calculated M indexes for each of the spots we represented with a special diagram. I will show it further. So this is the patient with paraproteinemia, with multiple myeloma in the course of treatment. So here before the treatment, then after part of treatment, next course of treatment, and next course of treatment. And you can see that if we will represent this index with this diagram, really good visible that after the third course of treatment, index of M gradient was increased. We put attention of the hematologists that it is necessary to do something because treatment now is not so effective as it was. And maybe after next course, paraproteins, quantity of paraprotein can characterize the mass of tumor in most cases, more cells, more immunoglobulins, and index allow us to fix this in the special significances. We proposed for hematologists change the treatment. Treatment of myeloma usually is chemotherapy, so another preparations were used for the treatment of the patient. And after the change of treatment, 3 months later, you can see the index of ingredient again decreased. So this change treatment, was effective for the patient. So this is illustration how we can use determination of ingredient indexes with the help of our elaborated program for the molecular control of effectiveness of the treatment, at least in this pathology. It's not so rare. Myeloma disease all the time is in between 20-25 tumors diagnosis which are reasons for death in Russian Federation last 5-7 years. One of the highly represented hemoblastosis, paraproteinemic hemoblastosis, it's a multiple myeloma. We must say that for the elaboration of the method of molecular control, we participate in the special competition on the between scientific organization and educational organization, and in 2015 obtained the first prize on the platform oncology. This is a diploma that we obtained, and actually we are actively publishing the results of our investigation, and to show that there is a request for our investigation in the world. I want to tell you that our publications are more than 850 times cited by scientists from the more than 50 countries of the world, between them Australia, Austria, Nigeria, Belgium, Brazil, Great Britain, Vietnam, Germany, Greece, Denmark, Iran, Ireland, Spain, Italy, Canada, Kenya, Cyprus, China, Colombia, Korea, Macedonia, Malta, also from Mexico, Myanmar, Netherlands, New Zealand, Norway, Poland, Russia, Serbia, Slovenia, USA, Taiwan, Tajikistan, Thailand, and Turkmenistan, Turkey, Ukraine, Finland, France, Czech Republic, Switzerland, Sweden, Estonia, Republic of South Africa, Japan. In more than 10 languages, the works of our department are cited, and in the special scientific libraries, which are measuring different coefficients and measuring the results of citation. The staff of our department have great indexes. For example, here you can see the Scopus index of our professor and chief of the department, Peter Konstantin Alexandrovich. It is one of the highest not only in medical academy but also in the whole Crimea Federal University, one of the highest index. Thank you for your attention. Now you can understand the significance of our coat of arms. Here you can see the immunoglobulin, but really these green parts, they indicating something that is interacting with the immunoglobulins, maybe complement, maybe some physiological components, but really you can see the objects that we are using as a source of model molecules for the investigation of immunoglobulins. Be in health. I wish you happiness and good luck on the exams. Goodbye.